Good morning. My name is Cruz, and I am an assistant professor at Purdue University. This recording is part of the APS Plant Health 2020 online meeting. And today we're going to be talking about some thoughts about the future of plant disease surveillance and pathogen diagnostics. Attendees at the end of this presentation will be able to understand several perspectives of past, current, and future approaches in epidemiological surveillance. I will also be presenting some preliminary results on the potential use of image-based data for epidemiological studies. And Dr. Mohammed Arif from the University of Hawaii and at Manoa will be talking about genome-informed technology advancements, especially about RPA and genome sequencing-based technologies that can be used for detection and surveillance of plant pathogens. And talking about plant pathogens and diseases, we know that they are a threat to crop production, and collecting and analyzing information associated with the epidemics caused by these pathogens is imperative and definitely important for successfully managing these diseases. And when we talk about surveillance, we need to think about three objectives: detection, estimation, and targeting. Now, we know that there are multiple challenges in early detection and quantification. One example is of logistical nature. This is especially true for row crops, for which vast and heterogeneous landscapes are often present. And one of the potential solutions that several authors have proposed is the use of advanced platforms that can be used for fast collection of big data. Now, when we think about big data, and this is more associated to row crops, so we can think of several types of information. And in many cases, we're thinking about heterogeneous data. But the idea here is to collect all these types of data and transfer them and also ultimately fuse the data for the purpose of building models. And so we know that models can be very helpful for making decisions. And in our particular case, we have been testing new forms of disease measurements to understand what they can tell us about the dynamics of disease epidemics. And so in this particular case, we had used tar spot of corn as a model system. So we have actually been working with microplots, and we have been collecting various types of data from various treatments. And particularly, we have used visual assessments, which is the traditional method for data collection. That data has been processed, and so we now understand the dynamics of those epidemics. But then we had also taken advantage that we had established those experiments for the collection of other type of data, including UAV-based imagery. That imagery and the information coming from that imagery has been processed, and we have obtained vegetation indices. And with that information, we have conducted various types of associations and correlation analysis. And at this point, this is just one piece of the puzzle. We are having some results that are intriguing. And for example, rates of change, including the rates of disease change actually coming from the visual information, but also we're finding some associations with the rates of change coming from the vegetative indices. And on another project, we have also developed a couple of algorithms that are capable of extracting information related to the signs and symptoms from images, and these are RGB images. So one of the algorithms is based on mask RCNN, and the other algorithm is based on contour analysis. Both algorithms have been capable of extracting information, and we have also been comparing the results coming from visual assessments and also from the extraction of the similar data from by using these algorithms. And very quickly here, some preliminary results and conclusions. So tar spot visual assessments were correlated with certain UAV-based phenotypes at various levels. Population growth models adequately describe disease progress for visual estimations and VI-based measurements. And as I had mentioned before, so from the two algorithms developed, actually one had a better performance, and one of the limitations of that one is that it's not automated, and we're planning to work on that in the near future. This is not the end, this is just the beginning, and additional research is needed to determine thresholds, understand pre-symptomatic relationships, and at some point start exploring the fusion of data, and perhaps including weather as well. I'd like to thank our collaborators, and also would like to mention that we have additional information for those of you interested in reading more about what we have done. We have also videos recently recorded. But in any case, so if you have any questions, here is my contact information. Please feel free to reach out. I'll be more than happy to answer your questions. With that, thank you very much.